can't get COMSOL to simulate a complex tensegrity, only a triangle. It'll only do a triangle. Not sure what's up with it.
Oh, okay. Okay, Bradley, Ken, Shannon, and I finished that paper.
Okay.
So it hasn't been submitted yet. I could do a presentation on it, I guess, well, when you're ready.
Yeah, that sounds great. It sounds great. Why don't you go ahead and do that?
Okay, no, not today.
Okay.
Maybe next week.
All right.
Okay.
So what paper is this?
This is the paper on reconstructing an image from a set of lines across it.
Oh, okay.
Oh wow, you've got that.
Yeah, it works.
Wow. Okay.
It's amazing. It's a prelude to another paper where we make the lines X-rays. But right now we're just trying to use lines.
That's great.
Okay.
Yeah.
So, okay. I'll put together something. We have 90 pictures. I have to reduce them for a presentation.
Oh yeah. Got to select those.
Okay.
Yeah.
Well, at any rate, it was fun working with him. He's straight out of high school.
Okay.
He didn't know anything about image processing, and he learned, and he knew some Python.
Okay.
He learned how to do image processing in Python in 3 months, I guess, and was able to come through with some fairly complicated stuff. Really pushed his high school algebra.
Wow.
So, but he's in college now, so that's the end of that collaboration for a while.
Yeah.
Oh.
So yeah, so that's great. That
Anyway,
sounds. Yeah.
Yeah, it's based on a strange observation, and that is, if you look at X-rays, they come out either in a cone beam, or if you collimate them into a fan beam. And there are thousands of papers on how to make images from them. But there isn't a single paper on how to make an image if you go to a one-dimensional beam.
Right.
Okay. So, at any rate, this will be the first.
Yeah. So it looks like. Oh yeah. So
Yeah, it's very strange.
yeah.
Susan might be able to turn it into some tensegrity structures. I don't know.
Yeah, if they would work. I was told to use MATLAB because it's just very straightforward. You're using matrices, and you can kind of see what's going on. So I'm going to try that next.
Next, okay.
Yeah, I've kind of got some images of what COMSOL produces, and then I have some other papers about other programs, and there's even some online that work for modeling metamaterials, and then modeling tensegrities and other things, and one's called Grasshopper. Anyway.
Okay.
So I don't know.
Look up. I just recalled the name of it. There's another program which I think is free. It's called Push Me, Pull Me.
Oh, that's it. Yeah. Thank you. Yeah, I was going to ask if you could remember that for me.
Yeah, just.
I'm having trouble getting hold of that.
Well, I haven't had any experience with it for a couple of years, so it could be that it's not available anymore.
Well, I ran into roadblocks. Like I can't download it if it's on, what is it? Google, Gmail, no, Chrome. Yeah,
Oh, yeah.
so Chrome won't download it, and then other things just, well, it just bogs. I can't access it.
Huh.
Oh, that's
somehow it's blocked.
Try Brave.
Pardon?
Have you tried Brave?
Brave?
Yeah. That's what I'm on. It's BRAVE. It's another what do you call it?
It's a web browser?
Yeah.
Huh. Okay, well, yeah, I'm having trouble with, what is it? This other one, Microsoft Edge, that's it. Won't let me into it. So I don't know if it's because it has is JavaScript. And I downloaded the Java and I don't know.
Huh.
Anyways, not working. Can't get into Push Me Pull You, which is specifically built to do tensegrities, which is probably what I need to try out my models.
Yeah, you got an example right here. Vahid is trapped in a tensegrity structure.
Yeah.
Actually, this is something trash.
You're not trapped? There isn't a big spider about to eat you, huh?
It's kind of something like sleeping, you can say. This is
yeah.
I'm in the southern part of Iran, and it's the weather is somewhat hot during this area, and I'm in a balcony right now.
Okay.
Yeah, well, here it's only -20. Not our problem.
Anyway, yeah, that looks like a nice structure behind you.
Yeah, it's got all the features, stiff and flexible parts.
If you are interested, that's what it looks like.
Oh yeah.
And if he tries to escape, you'll see the forces.
I'll be happy to.
Great. Yeah, so
anyway, I'll shut off my messy room.
Oh yeah.
Here, because it helps with bandwidth. So.
Okay. Yeah.
Yeah.
Yeah. So welcome to the meeting. This is the first meeting of 2024. So I hope everyone had a good New Year. Holiday season, and got to rest a little bit. And so yeah, I'm glad Susan's getting back into the swing of tensegrity. Sounds like a lot of challenges coming up on that. And I'm glad Dick was able to.
It's simply a headache when you try to use something like COMSOL with it, because it won't compute, won't settle down. It says that if the iteration error is less than 10 to the -14, then it's stable. And the graphs of the error show that it is below 10 to the -14, but it won't settle down. So, yeah, I don't think I either have to change the formulas in COMSOL so that they will do tensegrity, or I need to find another program.
Right.
Yeah, I think that's my conclusion.
Well, it sounds like fun. Yeah, I'm glad to hear it. Dick has got that problem in hand. We talked about that last year. Oh, he's been looking at it for a while, actually. So
I've been looking at it for too long, and I was more or less ordered to quit using COMSOL and do something else. And I'm going, can I quit now? Right now.
Yeah. So yeah. So welcome, Vahid. How are you doing this year?
Yeah, I'm fine, and Happy New Year to you as well. I hope you have a great year this one.
Yeah.
Actually, I was going to, because this is my second session I'm coming in a row, I guess. I was going to ask if there's anything I could do I could actually participate in and have maybe to take part in your conversations every week. with a better background, or if there could be anything I could be participated to, I would be happy to let me know.
Okay. Yeah, I mean, there are a lot of things that we're doing on a regular basis. It's just whatever interests you. We haven't done an overview of things in a while. So there are a lot of things that, I want to make it worth your time, to do things. But I'm not prepared to launch into a big presentation now about like what all things we have outstanding. But yeah, I would definitely like to have you you could even like just present on what you're working on I know you're working on some things with, ion channels and things like that, or other things that you're working on. You're free to present that too. It might fit into something we're doing. So.
Okay. Actually, as you said, I've been more focused on the ion channel models during the past years in the OpenWorm project. But recently, I've been more trying to focus on a broader approach to network and system models. But on the other hand, because I'm actually trying to have an evolutionary actually point of view to what I'm working on, I would be happy to look at the different stage of the development of the worm. One reason is that I'm more interested in to see how the brain of a worm could develop during different stages, and how it could become more complicated from some simple I can say from an engineering point of view, because I have some background in engineering as well. Let me say from the beginning, I have a bachelor in computer engineering. I've studied a master in biochemistry or biology with focus on biochemistry, and during my thesis I was working on the OpenWorm. Then I started another. Actually, this first master was going to be continued in a lab after some discussion we had in one of the labs at Stanford, but it didn't happen at that time. So I started another master in mechatronics over here again. My second master in mechatronics was mainly focused on the electrical circuits, and how they could be, and different applications of our simulation models from an engineering point of view. So I'm so interested now to see how a simple model of neural networks in a simple brain, like maybe we can say at the very beginning of the development of a worm like C. elegans, could be somehow be developed to be a more complex one, like a C. elegans in 20 days. And after that, what will happen if it is actually is going to be more complex, maybe some other animals and the humankind. On the other hand, I'm somehow interested in the application of those models from, we can say, in different diseases, as I said, like channelopathies, like a lot of diseases that ion channels especially are included in. And there are two main, I can say, studies these days I'm more involved in, and would be happy to co-study with you, collaborate. Anything you are actually doing, you are maybe I'm seeing that you are so active in reading and studying different papers every week, and maybe summarizing those papers. I would be happy to collaborate on that as well, and any other suggestion that could be possibly by you.
Yeah, that sounds great. So yeah, we have an interest in doing things with like the development of the brain and things like that. That would be great to focus on to see if you could gain some insights from
Yeah.
that.
And
Uh,
one more thing that I've been actually looking into was a comparison between artificial neural networks and biological neural networks. And this is something that is really interesting from an evolutionary point of view, and also from a developmental point of view. As you know, when you uh, try to analyze or build some electrical circuit from the beginning, you can get a lot of idea from how it has been developed from a biological point of view, and this is something that already happened during the development of artificial neural networks. So I'm a little more interested in that topic, focusing on that topic with your collaboration maybe. And yeah, and like I say, and one more thing that these days I'm also looking for a PhD program that maybe I could be more get focused on all these topics. So I hope during the other 4 to 5 years I would be more focused on all everything I've been discussing right now.
that sounds great. That sounds great. Yeah, there's a lot of space for that. We actually I worked on a paper with someone who used to collaborate in this group. We worked on a paper on comparing artificial and biological neural networks. I have to dust that off and show you because it's not, and it's kind of like you can make a point by point comparison, but I think there's something deeper there. Like there's some principle or something that maybe biological neural networks are better at or are different from in the way that they work that makes them I mean, it seems like in artificial neural networks we hit this wall of understanding and things like that. And so I mean, why is that? And so why are the networks in biological neural networks the way they are, and why do they work so well and things like that? So yeah, I would definitely also, if there are papers you'd like to review on the topic, because the reason we do that in the group is because it helps, I think, people think about, some of these papers, sort of, where do they fit into a lot of what we're doing, and so yeah, please feel free to present on something if you want to bring it to the meeting, and just, it doesn't have to be formal; it can be just very informal.
For sure.
Yeah.
But if I know every week we are going to discuss any specific topic, I would be happy to collaborate with that topic as well.
Okay.
I'm not sure. I actually do not have any agenda or anything like that for weekly meetings. So if there is any specific topic that every week you are going to discuss, I would be happy to read some paper and discuss on them as well, or summarize something like that.
Yeah. Yeah, I usually just leave it up to where it goes. Like, I usually have some materials, but then I see where we're, what fits into the week, or it doesn't even matter because we can just present on something. A lot of times, like we'll do, during the Google Summer of Code period, for example, we'll do like presentations on like machine learning and deep learning, and then we'll switch over to like biology, or biophysics. So it's not, it doesn't need to be necessarily match the theme, but we do want to we do want to get a mixture of things because it is valuable to see different perspectives in.
For sure. Yeah. And one more thing to focus again to emphasize, because I've been somehow more active during some years in the past in the OpenWorm project, and a little less active during 1 or 2 years ago. But I was going to emphasize this again, that I'm going to focus on my studies from I will start from this year, and it would be continued for maybe 4 or 5 years, because in the past I've been somehow I was more a little busy with my job, I've been both working and studying as well, but this year I definitely stopped working, and I'm just going to focus on all my research and studies from now on a PhD level. It's my decision of last year, but I'm going to continue for 4 or 5 years. So you can count on me for the next 4 or 5 years, definitely.
Yeah. Well, that's great. Yeah. Yeah.
So I'll be happy to enjoy.
Yeah.
Yeah.
Yeah. So yeah, if you need anything, let me know. I mean, if you want to coordinate something, or if you want to do something, if something really kind of gets you really interested, we can do some, we can develop it. So.
Yeah, I would be happy to, and especially if my next maybe thesis would be on something like some similar topics, or with the collaboration of some supervisor from the worm community, from the C. elegans community. That would be really would maybe match everything we are going to do. So it a little depends on what I'm going to work, my focus and my supervisor, but definitely I would do some similar topics as I said for the next maybe 4 or 5 years.
Okay. Yeah. Yeah, it's definitely good. I've had a couple people do things like that where they've kind of developed things in the group and then gone on to work on them for like a graduate project or something. So yeah, definitely it can always tie into that.
We'd be happy to collaborate.
Yeah.
Okay.
All right. Thank you. Yeah.
Thank you for the great time you are actually devoting.
Yeah. So we had someone else, David. I don't know if they were just checking out the meeting or what, but they haven't joined back in, so I don't know. So yeah, that's great. So I guess.
Actually, sorry to finish.
That's fine.
Topic.
Okay.
If you know of any specific program that would be related to everything we are talking about, I would be happy to apply for that program as well. Because these days I'm applying for 1 or 2 related programs for MS plus PhD program, and so if you know of any, some supervisor with some maybe similar topics that we are discussing, is looking for a researcher, a C. elegans researcher, I would be happy to apply for that program as well. Yeah.
All right.
I guess this is the last thing I was going to discuss. Maybe I have finished my points.
Okay.
Well, you could always join APS Physics. They do biophysics and soft matter physics, and sometimes they have workshops, and sometimes they have postdoc positions that they advertise, and you can get to know the community a bit if you join them.
Thank you. Could you write in the message? Yeah, in the message.
Oh, sure.
Yeah, thank you. And actually, last week I applied for a mentorship program that I saw a C. elegans community has been greatly working on. Yeah, there was a list of different mentors, and I saw your name under the list.
Yeah.
The only one who was actually focusing on computational neuroscience of C. elegans, actually computational biology of C. elegans, and if there is anyone, I hope if there is you or anyone else could be a mentor of the work that you're working on, I would be happy to collaborate. I registered my name under that list as well, so I'm hoping I will finally find someone supervising my studies more professionally there.
Yeah. Yeah, I think this is something that I signed up for like a couple months ago. I didn't know what the state of it was, but I'm glad that it's happening and that You're fine. Yeah, I guess, the C. elegans community is really, outside of OpenWorm, they really have a lot of good opportunities for open data and mentorship. And so, it's like, it's pretty well, pretty tightly knit, or I wouldn't say tightly knit, but like it has a lot of resources. So, yeah, definitely, participate in like a mentorship program if you can. And I'll get in touch with you about some of the things. I've been taking notes about some things that you might be interested in. I'll send you some things on that because we've done some work on developmental connectomes, and there's a fair amount of work on that already. But it's interesting because it's a relatively small nervous system that's well characterized. But at the same time, there are things going on there in development that we don't really know. What how it works or how it gets connected and how it gets put together. So I think there are a lot of open questions there. It's just a matter of what kind of data can we get, and what kind of questions can we ask. So there's in C. elegans you have larval development, which is where we have embryogenetic development, where we have the cells divide and form this worm, and it's all curled up in the egg. And then once it hatches, you have larval development, which is where it goes through a number of stages, molts of the cuticle, and there are a lot of changes there as well, a lot of morphogenesis of cell types, and so it's interesting. There actually are some data sets for larval development for the brain, and those are pretty useful. For the embryo, not so much, but the embryo we have a lot of data on cell differentiation for neurons, and so that's where it's important. Important data for that. So yeah, go ahead.
If you remember, we were discussing it. Some idea in the past I was working on. There was an online book I was going to work on called Worm Math Book that I was going to somehow discuss the different stages of the development of a worm from day 0 to day 21, something like that, and how it evolves through all these states from a mathematical point of view. If you, for example, design in a simple, you can say in a simple form, if you design an electrical circuit that could handle some very basic actually behaviors of a worm from day 0, how it could actually evolve during all these days and become more complex and could decide what to do and everything. Let me find the main idea of the repository that I'm actually working on. I have some references that I'm day by day adding to that book as well, to be able to maybe put it in a way that could be collaboratively work on that as well. I guess if that could happen during maybe the next years that I'm going to work especially with help of someone who is really maybe who has a great background in both mathematics and the biology of C. elegans, would be really great to work on this as well. This is one of the ideas that I'm, especially from a comparison point of view between ANN and BNN.
Oh, yeah.
So that would be a great case here to work on. I would be happy to work on some similar matters as well. Yeah.
Great. Yeah. It's actually the scale of C. elegans development is like hours. So days is the whole lifespan is like 3 days. But yeah, it's definitely a good problem. You can break it down because the data exists. People have done the characterized everything. So yeah.
I just wanted to say something more about the APS March meeting. The early registration is now open, and I know that I'm going to get this name wrong. Marchetti group is presenting. Like they're the active matter, they're an active matter group, is presenting quite a few times at the meeting.
Yeah.
And there's some workshops that look interesting as well. I don't know if I'll go to a workshop, but I was thinking of attending virtually. I got a lot out of it.
Yeah, last
year, the year before. And when I attended this past year in person, it was in Las Vegas, and I got sore feet and sat in meetings and took hasty notes, and it was almost better to do it online where you could do occasional screen captures and then you can look things up. So it's okay to attend virtually. It's quite nice to do that. It's cheaper and easier. You don't get the interaction with people, but still. Yeah, I attend the Biophysics students group meetings, and I got to meet those people in person. So it's nice to meet people you've met online. And some of you I would like to meet in person someday.
Yeah.
Yeah.
Great. And is there anything related to the quantum phenomena because I really that's a part of it?
They have quantum physics as a part of the APS physics. I don't know if it's related to biology, but yes, it's all of physics that's represented by the APS physics meeting.
I mean, because as you know, some quantum phenomena are recently they have been discussed that we have especially in ion channels. I've read some papers that quantum phenomena could affect the maybe somehow the pass of ions through the cells, through the membranes, and so I was really interested these days in see if there is any we can say really strict study in that topics. If they are going to discuss something like that as well, that would be great.
Yeah, I don't know know about quantum wells in transistors. So I'm sure there's an effect in biology, and it may have something to do with Brownian motion. To go in that direction. So yeah, they have all kinds of things at this APS physics meeting.
Yeah.
So just look it up. It's like you certainly can't attend it all.
Yeah.
Sure.
Yeah, we had some interesting things last year. You had presented on some of it and talked about it, like what you had found interesting. So yeah, definitely a lot of soft matter physics. And then I think that is really kind of quite an interesting set of questions there in soft matter physics, and especially with respect to development. And it's sort of a hot topic now where people are finding these different questions, of kind of like, if you have cell division, you have tissues that are forming, they're forming sort of soft matter types of, they can be treated as a soft matter, and, you can then model the properties of that soft matter as sort of like getting a sense of what the tissue is like, how it behaves, how it deforms, and then that explains certain things about development. So, but that's something that is an emerging area. I imagine I really haven't dug into the math of it, but like, you get to apply a number of different models to, an embryo. So
I think I'm just going to go with the vertex model for my epithelial tissues. So people have already done a lot of work on it, and just try to use something that's out there already. because it seems to work.
Yeah.
And you don't need the whole cell. You can model the whole cell in order to get when tissue acts. It's,
yeah.
It's a vertex model and does behavior very well. And for epithelial tissue.
Yeah. Yeah, we've talked about the epithelial. Well, I mean, in a number of papers we did last year, we talked about epithelial networks and things like that. So.
Yeah.
Yeah.
Well, brain is epithelial tissue.
Yeah. Yeah. So yeah, it's good stuff. Yeah, I'll get together some things for Vahid, and you can take a look at them and see, and we can just shape. Usually we just kind of work through things from week to week and see where we are after that. And so great.
Actually, the link I was following right now for the APS, do you mean this is the main work you were talking about?
Sister?
Is this the link of the meeting that we were talking about?
Yeah, this is APS.
APS News.
This is
okay. I like D-Bio better, but
yeah.
soft is, I also want to be soft.
Yeah, this is something that maybe, as they are describing, that quantum phenomena is not affecting, in contrary to what I was talking about, because these days I've been talking to some people in the biophysics world, and especially the neuroscience world. I've been talking to Dr. Peter Dayan in the past, and we were discussing the different type of maybe approach to the models of how neural networks work. And one thing I've been discussing was the effect of the quantum phenomena that some people are discussing during the past years. And actually, this is where we're going to. I've been decided to even to apply for a biophysics program these days, in maybe for the next program I'm going to join, and maybe in that context I would study better the effect of quantum physics in the world of neuroscience, and this is something that would be really interesting, I guess. But
yeah, I'm sure there's pros. I'm sure there's arguments for and against. I mean, there's arguments for and against tensegrity as a model system for cells. So,
at least we know somehow that the probabilistic, maybe we can say the probabilistic theories that are somehow working both in the quantum world and in the mathematical models that we are using, maybe that could be a good starting point to discuss something like that, because lots of mathematical models we are working on are somehow are working from both points of view.
yeah, there's even a statistical physics chapter that I joined because they wanted me to. They had the T-shirts, so I tried to win a T-shirt. I got a tote bag instead, but I joined the statistical physics chapter.
Yeah. Well, that's great. Thank you for that discussion. Looks like so. That's the link to the Soft Matter group, and yeah, we'll talk about this more in weeks to come. And yeah, we'll flesh out some things. Looks like Morgan and Jesse joined us, so welcome. Hope you had a good break. I'm gonna share my screen. I want to talk about some things that kind of collected. I know Morgan sent me this interesting collection. I want to go through that, and then I want to talk about a paper. So let me share my screen here. So this is something Morgan sent. This is a science special issue on what they call the Brain Cell Census. And this is another thing we've talked about in the past about like cell types. And we talked about that actually at the last meeting of 2023 about cell types and cell atlases and like, getting a good approximation of the number of different types of cell. And, you can do this in a number of ways. You can estimate them from, microscopy observations. You can do like next gen sequencing, profiling, things like that. So there are different ways that people are kind of coming at this problem. And I guess the answer would be to like use a bunch of different methods and get like a fair consensus around like what is the number of different types of cells. What they're doing in this collection of papers, this is recently published in Science in November 23. So this was a couple months ago. What they've done is they've done this brain cell census, I think in humans. So, the human brain has 86 billion neurons, and they want to decipher the genetic diversity of these neurons. And so when they do that, they can actually then put them into different categories of, functional category or just kind of identity category. And so the difference, of course, is that the functional categories are, you can look at functional genes, you can look at just differences in genetic makeup, you can look at different things like that. So this is how they're, kind of approaching this problem. And then, they come up with some sort of estimate of the different distributions of cell type in the brain. So we have under this, what this link was, and I think Morgan put this in a Slack as a Slack link. This is 21 papers. So this is in this collection of papers published in Science, Science Advances, and Science Translational Medicine, so all the science journals that they have now. The National Institute of Health's Brain Research through Advancing Innovative Technologies, or their Brain Initiative. And then there's the Cell Census Network, BICCN, which is a part of that, presents a large-scale multi-omics. And multi-omics is using these different genomic or omic approaches to looking at usually gene expression, but with omics, you sometimes also get the sequences, and you can take those sequences and map them to like a draft human genome and or personalized human genome and get figure out what genes those are, what loci, and then you can identify cell types that way. So they present a large-scale multi-omics analysis of the human brain at the cell type level. So they're looking at different cell types, they're looking at the diversity within the cell type. So you could have a conventional cell type like a cerebellar, different cells in the cerebellum. You could then look at the cells that are known to be of that category and look at, what they're expressing, what genes are there that are, being regulated in different ways and so forth. And so not only can you find category or, do this sort of taxonomic exercise, but you can find diversity within different cell types and in ways that we can't really do with anatomy or with microscopy. So this is what they're doing here. So they have 21 papers. They're producing a comprehensive cell atlas of the adult and developing human brain at the transcriptional, epigenetic, and functional levels. In addition, by comparing human, non-human primate, and rodent brains, so they have human brains, but they also have non-human primates, and they have rodents, so they have this sort of phylogenetic diversity. And, this is, of course, I don't know how much people think about this in terms of phylogenetics. A lot of times in these kind of studies, they just make the comparison and leave it there and say this is the difference between rat brains, say, and human brains, or mouse brains and human brains. And so that's not that interesting in and of itself. Of course, there are going to be differences. But what are those differences? Where do they diverge from? What does that mean functionally and so forth? So that's not, that's good that we have these different different species models that we can look at, look across. And so this is one thing that we talked about with respect to cell atlases last time, is that cell atlases don't give a good accounting of, say, like lineage trees or, differentiation processes or evolution or anything like that. They're usually just kind of identifying things, from the observations and maybe doing some sort of classification exercise where they're just saying, this is this type of cell, and it differs maybe from mouse, which is a lot of times what maybe say like a neuroscience, if you're doing clinical neuroscience or just sort of behavioral neuroscience or something like that, that's what you're interested in. So that's maybe one reason why that's a priority. But, that's one shortcoming of cell atlases, as we talked about last time. So until now, this type of analysis is mostly reserved to preclinical studies and animal models. The results presented by the BICCN show how these technologies can now be applied at scale to tackle brain complexity in humans and their close relatives. These data will allow researchers to tackle a wide variety of scientific questions and are already a milestone in the neuroscientific world. So this is kind of getting at these, fundable questions, as they call them, or fundable statements, which is they kind of give the significance in terms of not, maybe not practical significance, but it's like, the sort of thing you might think of when you kind of develop something. You don't really know what it is. You just kind of say, this can improve the world or something like that. So there are a number of papers in here. This one, single cell genomics reveals region-specific developmental trajectories. So this is where they're looking at different trajectories of different parts of the brain. And so they're looking at neuronal diversity in the human hypothalamus. So they're looking at different parts of the hypothalamus, which is a part of the brain. They're looking at single cell genomics, and they're trying to figure out, why there are these different parts of the hippocampus and why, how they differentiate. They don't really, I don't know if they get into the question of differentiation, however. It's just kind of identifying that there are these differences and then maybe the trajectory to those. There's one, the single cell genomic atlas for maturation of the human cerebellum during early childhood. So this is a developmental study where they're looking at the maturation of human cerebellum. They're looking at transcriptome diversity of cell types across the adult human brain. So they're looking at how, gene expression varies across different cell types. Comparative transcriptomics, which means that they're looking at the transcriptome or how genes are expressed in different species. This reveals human-specific cortical features. So it's human-specific as opposed to, say, like whatever they're comparing it with. It's still not really a statement of evolution. It's just a statement of difference. So if you want to know something about the evolution, you might want to do like some sort of look at the trajectory of some area that's unique to humans or, whatever, and then go from there. So, putting these together, I think you can see that, any one study isn't going to cover really what we would like to see maybe in our group, but they're kind of, we're putting the pieces together here. Let's see. This one, morphoelectric and transcriptomic divergence of the layer 1 interneuron repertoire in human versus mouse neocortex. So this is a comparison of human and mouse. This is the specific area, layer 1 interneuron. This is, I think, in neocortex. So this is the, neocortex in mammals. It has 6 layers, and this is layer 1. And so layer 1 then has, different properties in human versus mouse. And you can look at that in these two ways. So you're just taking a part of a tissue, a part of a region of a tissue type, and you're looking at the cells there and the diversity and how they differ between species. This one's single cell analysis of prenatal and postnatal human cortical development. So this is where we have, these 2 time points, prenatal and postnatal, and then you look and compare what they're expressing. And so, in C. elegans, the difference there would be like if you were to look at, say, like a larval stage, or in the embryo, in the in the embryogenetic state, versus maybe in the adult worm or some other maybe later larval endpoint. So you have 2 endpoints and you're comparing. And so, the idea here is that you would get a sense of what changes with development, what's going on in development, what's going on post-development. And so it's supposed to give you some indicator of that. So yeah, there are a lot of stuff on development here, a lot of stuff on individual variation. So this is where you look at a bunch of humans and you see what the differences are between cell type abundance and expression. And so, you can do this in other species as well. In C. elegans, we can do this with different defined genetic strains. So, there is data out there in C. elegans for different defined mutations, where they have maybe a mutation for brain development or a mutation for like movement deficits. And you can actually get a whole catalog of these defined mutants. And there's a lot of data out there that's been generated on these defined mutants. So, being able to compare that with a wild type allows you to see like where these differences, if you knock out something important, like a certain gene, and it affects the phenotype, what is the difference between the two? It's not something you can necessarily do in humans, but in something like C. elegans is much easier to do. But anyways, that's the basic type of experimental comparison you have. So yeah, there's a lot of stuff here. It's just, a lot of good papers. Basically in that region, we have just atlases where we have a cellular resolution atlas of Broca's area. We have different types of profiling. We do they're doing a lot of stuff with development, with, diversity of neurons, and kind of getting that space nailed down to some degree, so that's the brain cell census. And I'll send a link out after the meeting on that. So yeah, thanks Morgan for bringing that to my attention. Is there anything you'd like to say about that, or?
Well, I'm just about to head off for a school run.
Okay.
But yeah, a lot of good papers to take in there, obviously.
Okay, so then the last thing I wanted to do was I wanted to go over this paper here, and this is a paper out of a new journal, PRX Life. So this is Physics Reviews X, and they have like Physics Review X is like the open source version of Physics Reviews. They have like different letters after the Physics Reviews, and this is X. And then they have this new journal called PRX Life, which is focused on biophysics and biology, and from a physics perspective. So there are a lot of interesting papers coming out in that journal. I think they've had like 2 issues so far. This one is from the first issue. This is models of cell processes are far from the edge of chaos. And this is a number of authors on this. So the abstract reads, complex living systems are thought to exist at the edge of chaos, separating the ordered dynamics of robust function from the disordered dynamics of rapid environmental adaptation. So what it means is that you have a biological system. First of all, it's this complex system, and it exists at what they call the edge of chaos. And so the edge of chaos means that it's not like a deterministic system. But it's also not a chaotic system. It sort of exists at the edge of what they call chaos. And I'm not going to go into like what physicists mean by chaos. It's different than what the colloquial term is for it, because we usually think of chaos as highly disordered. In physics chaos, things are not necessarily highly disordered. but they're highly variable, and there's certain properties to chaotic systems that are, being at the edge of chaos is important for leveraging those properties, but also having a system that is, functional under a wide range of conditions. Things don't break down necessarily, but they become, adaptive. So that's why they're separating the ordered dynamics of robust function, meaning that you can have like a pathway that operates under many different conditions. So like, your metabolism operates under a wide variety of conditions, and that's a robust function instead of being a brittle function where, maybe your metabolism worked like a machine where it might break if it, gets out of whack, if the parameters are, pushed. Like if you get sick, for example, you don't want to die. You can tolerate a lot of like ill health before you die, maybe. And so that's what robust function means. And then that's compared with the disordered dynamics of rapid environmental adaptation, which means that if you need to adapt to the environment, you can, but that also introduces a lot of disorder to the system. So this is what they mean by edge of chaos. So here, a deeper inspection of 72 experimentally supported discrete dynamical models. So these are just dynamical systems models that are discrete. And this is of cell processes. So they have these different models of cell processes. Reveals previously unobserved order on long time scales. So they're looking at long time scales, simulating them over long time scales. And there's this order in the system that they're identifying. And so this suggests a greater rigidity of these systems than was previously conjectured. So this is so basically we go back to this idea of the edge of chaos. The edge of chaos, we think about chaos, we think about it being disordered, we think about it being highly adaptive if it's on the edge of chaos. And then we look at the models, we do this study, and we find that there's this order on long time scales. So that means that when you have these kind of changes, you have this regime of sort of being change friendly, that it still kind of hews to this long time scale of order. So, a good example of that is your body, your metabolism runs at different sort of, you can adapt to a lot of things. You can get sick, but you don't die from certain perturbations to your metabolism. But your metabolism is still sort of has set points. It doesn't break down over time unless you die. Maybe when you're aging, it breaks down a little bit, but there's this underlying order. And so there's a greater rigidity than we kind of thought from the models. So this is an interesting kind of thing to think about, that we have these physiological systems, say, or a complex living system like a cell that has a wide range of sort of operating parameters, that the operating parameters can be pushed in many different ways in many different directions. And the cells or the system can adapt, but it also doesn't go out of state. It stays within a certain state. So this is really one of the things about biology that is kind of fascinating, is this sort of robustness over time. So we find that propagation of internal perturbations is transient in most cases. So when you perturb your system, it doesn't necessarily stick with the system. You might have a memory of that perturbation, but it doesn't really adversely affect the system. Now, it may be that, if you get sick, it might maybe injure you in some way, or it could be like, that if you get sick, that you build up a resistance to something, but that's not really what they're talking about here. In any case, there may be some counterexamples of that. And even when large perturbation cascades persist, which could be where you experience a very large perturbation to that system, and there are all sorts of things that result from that. So, if you have, like, say, for example, a large-scale environmental perturbation on a species, you might have, not an extinction, but a large die-off, that those effects are often minimal, but sometimes they can be fairly large. It just depends on what's going on in the system. But the idea is that there may be some memory for perturbations, but it doesn't predominate the system. It can operate in the face of a lot of perturbations and retain its essential character, as we would say. Moreover, we find evidence that stochasticity and desynchronization can lead to increased recovery from regulatory perturbation cascades. So this is where we have stochasticity, where we have randomness, and desynchronization. So sometimes things will become desynchronized to allow for recovery. So, if we have tightly interrelated biological processes, they don't exist as like cogs in a machine. They can be desynchronized. And I think a good example of that is like brain synchrony with brain states, that those can be desynchronized or synchronized in different ways that are adaptive. So this is kind of an interesting point as well. Our analysis relies on new measures that quantify the tendency of perturbations to spread through a discrete dynamical system. So this is where they're looking at these new measures to look at, like, if you perturb a system, a dynamical system, can we look at the spread of things throughout that system? And so that suggests, things like networks. One of the authors on this is Reka Albert, who did a lot of these seminal papers on network science with Barbacea, and so that's, and Luis Rocha has done a lot of stuff with ecology and other types of complex systems. So these connections are important, and so we can actually develop measures that look how things propagate through a network and spread through. We can model the network as a discrete dynamical system, propagate through that. Computing these measures is not feasible using current methodology. Thus, we developed a multi-purpose CUDA-based simulation tool. So this is something, it's a computational tool, which we made available as the open source Python library, CubeWalkers. So they actually have this Python library you can download and use for this. Based on novel measures and simulations, our results suggest that, contrary to current theory, cell processes are ordered and far from the edge of chaos. So they're actually making a different argument here from what most people are making, and this edge of chaos argument, I think, really has been like kind of sort of a qualitative judgment about cells and other types of processes that basically, they're at the edge of chaos because they look like they're very dynamic, but we don't know. They're obviously not chaotic, so they're at the edge of chaos. And so they're actually saying that that's not true, that cell processes are ordered. They have a lot of order, especially long-term order. And so while they're robust, they don't exist at the edge of chaos. And so they talk about the edge of chaos here. This is a coined term by Norm Packard in 1988. So this goes way back into the early days of artificial life research. And in complexity research, this idea has been around a lot. This refers to the tendency of adaptive systems to evolve towards a dynamical regime that lies between order and disorder. So this is a tendency of a lot of very complex systems that are very adaptive. You can think of cell processes, but you can also think of like urban systems or, other types of systems that are very complex, that have a lot of moving parts and actually look to the eye, which is important, that they have a lot of properties of chaos but aren't chaotic. So in systems biology, this is often referred to as the criticality hypothesis and is closely related to the work of Kaufman and Derrida, who demonstrated that simple tunable models of gene regulation exhibit an order to chaos phase transition. So this is where you have a model of gene regulation, maybe a gene regulatory network, and you can actually exhibit this. You can go from order to chaos, and there's this phase transition that characterizes this. And so near this transition, it is conjectured living systems optimally balance the rigidity required to function in a noisy environment with the flexibility required to undergo developmental, metabolic, and evolutionary processes that depend on cellular context. What that big sentence means is that basically you can balance the need to sort of be very adaptive if you're in a noisy environment, but you can also do that in a way that's flexible but also consistent with your mechanisms that you have available to you. So it isn't just a free-for-all. You don't just adapt in any old way. You can adapt to a lot of different conditions, but you have these mechanisms that you can use, and you can go in a certain direction, or you can return to your set point as needed. And so we don't really we've talked about edge of chaos for years, but people haven't really understood what that actually means in a very rigorous context. So this is really kind of going through. Actually, the Derrida study was in random Boolean networks. So I guess this is a type of, well, you can model GRNs using random Boolean networks, but this is where this is the kind of level of analysis that people have been using for this, where they have a random Boolean network, which is where you have zeros and ones, and you have this random network, and you simulate things on that network. So that's the level of analysis. In this paper, they use many, many different types of tools that are much more rigorous and complex than what they were using there. And so they actually have a Boolean network that they're using, but it's comparing different types of models. So they have this software, CubeWalkers. This is a Python library. This is a Boolean network simulator, and it allows you to. It supports various user update schemes, node and edge control interventions, and probabilistic update rules. And so this is where you can do this. They have different categories like cancer, cell cycle, development and differentiation, metabolism, and then they have different types of models, highlighted models like apoptosis network, cell cycle for Arabidopsis, cortical area development, signal transduction of fibroblasts. So a lot of different cell cycle transcription, a lot of different model systems that they have in there in different categories. So yeah. So I don't know where the link is, probably in the paper somewhere for this repository where the software is. But I'll finish with this, where they have a graph of this phase space. You have robust, sensitive to robust, sensitive, and then robust to sensitive. So these are short and long-term perturbation responses in the cell collective, measured in a phase and sensitive way. So this is where you have a robust response, a sensitive or robust response, sensitive, and then robust to sensitive. In the robust regime, lower left quadrant, both short-term and long-term responses are below 1. So that's for this Derrida coefficient, synchronization coefficient, which indicates perturbation extinction and is characterized by ordered dynamics. So this is sort of the ordered regime. It's robust, but it's ordered. In the sensitive regime, which is up here in the upper right, both short-term and long-term responses are above 1. This indicates perturbation growth, which means that it propagates throughout the system. The perturbation dominates, and you get maybe chaotic dynamics. And then the other two quadrants indicate cases of disagreement between the short-term and long-term responses. So this is where they're competing, and there's this phase transition between the two that you could characterize. In contrast with the traditional approach depicted in the left panel of figure 4, which is we're not going to see, this figure illustrates perturbation response when source nodes and phase shifts are accounted for. Basically, they're trying to model sort of the properties of this network where maybe the nodes have different states. And then most models show a substantially more robust perturbation response when these factors are taken into consideration. The symbols indicate the model categories, so you have these different symbols in here. So they have this ensemble of models, and they're modeling these different transitions, sensitive, robust to sensitive, or where the nodes have different states, and they sort of fight it out for, which one's going to dominate. Robust is where everything is sort of ordered, and then sensitive is where everything is chaotic. So now I'd like to go over figure 10 in this paper. And these are 5 different ways or 5 different systems in which they apply their random Boolean networks. But more importantly, they get the state transition graphs, which you can see here, to show sort of how there's an update dependence. So in other words, the previous state affects the current state and so forth. And so without getting into the measures that they use in the paper, we can see that there are 5 different examples from different, I guess, biological systems, and then the different ways in which these network topologies are configured. And these are the state transition graphs, so the different ways in which they're updated. And so it makes sense to have different configurations for different types of systems because different systems operate differently and have different dynamics. So the first one are delay sensitivities and oscillations. So this is, again, where you have a synchronous and an asynchronous state transition graph. You have where, this 0, 0 doesn't update anything, but 0, 1 leads to 1, 0, leads to 1, 1, and then back to 0, 1. So these 3 states here are transitive and 0, 0 is left out. In the asynchronous case, however, 0, 0 is the end state for 0, 1 and 1, 0. 1, 0 leads to 1, 1, and there's this feedback between 0, 1 and 1, 1. And so then this leads to this transition graph down here. And this is a 3-state system, or a 3-bit system, I guess, where you have different state transition rules. The second one is an attractor Bayesian update for the robustness of metabolic interactions in the gut microbiome. So this is where we actually take a specific type of system, not just oscillations, and we map them to this random Boolean network. So we have 3 parts of this, for the different components of the interactions. We have an order that we work from. We have the shared part of the state transition graph. We have the rest of the synchronous state transition graph, and then the rest of the asynchronous state transition graph. And then for the third example, update dependence in colitis-associated colon cancer, we have this state transition network here according to these rules. These are the different components, and then the different transitions between them. And then we have our synchronous state transition graph with the state order, so we use that order to build that graph, and then an asynchronous state transition graph with the same order, but then we also take asynchrony into account. Then we have update-dependent attractor basins and cortical area development. So this is where we have different genes, and we have a state order here. And then we have states colored by asynchronous basin of attraction. So they go from yellow to green, or yellow to green, and they show from synchronous to asynchronous. So there's this gradient, and then these are the different states and how they allow for these transitions from state to state. And then finally, we have attractor basin size in a simplified apoptotic model. So we have these factors here, Cas3, Cas6, IAP, and we have the different rules, transition rules there. And then we have our synchronous state transition graph with the state order based on this graph at the top, like we did for C. And then we have the asynchronous state transition graph with that same state order. And so that gives us an order for rules for these transitions. But we have both synchronous and asynchronous cases. If we look at the legend here, we can see that panel A illustrates delay dependency in the examples of different equations that they introduce, which are inspired by cell cycle transcription by coupled CDK and network oscillators. So in biology, we have this oscillator in the cell cycle that they're using as inspiration for this model. Panel B demonstrates how much the state transition graph of the metabolic interactions in the gut microbiome model, which they cite as citation 40, is robust to changes in update scheme. And we can use other systems like cortical area development models and a reduced version of the apoptosis network model, which is cited in 43. In each of these interaction networks, each node symbol contains the update function of the node. Blue edges ending in filled circles indicate positive regulation, so that's up here. And then the red edges ending in open circles denote negative regulation. these blue edges of circles are positive regulation. These red edges with open circles represent negative regulation. And the same holds true for E as well. In asynchronous updates, states can reach more than one attractor. So such states are shaded using a gradient, and so that's why we have these gradients in these two examples, C and E. In the lower part of panel A, the states can differ only in the value of the delay node Y and are grouped together in shaded boxes. So the shaded boxes are here, as you can see, they form sort of subregions of the state graph. I think that's an interesting paper. It kind of gets into like a lot of these things when we say the cell is very complex. Okay, but how do we look at how do we understand and characterize it? Okay, so any comments or questions about that paper?
I'd like to know more about the Boolean operations they're working with.
Yeah.
I don't know if reading the paper would help.
Yeah. Well, I think the idea of like a random Boolean network is that each node in the network has a state at 0 to 1. It can be flipped. So you could have like a network where you flip it from one state to another, ordered to random or some other set of operations, and then the operations are just under what conditions that flip happens. It could be probabilistic. It could be something where certain conditions are met, and so that's basically the idea of that kind of network. And a random network just refers to how it's connected. So the nodes are connected in a certain way. Random networks are usually like, connected randomly. You could have scale-free networks, which are connected in different, to different degrees, and other types of networks as well. So, yeah, then the way it's connected, of course, depends on how things propagate through the network. So if you have a node that's like, disordered, but it's not very highly connected to the network, its effect on the network as a whole is going to be decreased because it can't really get its message out to the other nodes. And, that, and the converse holds true as well. If there's a highly connected node in the network, and in random networks, you don't really have this, but in something like a scale-free network, you do, or a small world network, especially. If it's very highly connected, that node, if it's disordered, can really broadcast throughout the system and really kind of have an outsized effect. And so the way that you organize the network is important. The conditions under which the nodes can be switched from ordered to disordered is also important.
Don't know if it would work for a tensegrity network.
Well, I think like a tensegrity, your connections are in this kind of regime. It's more like the connections are sort of physical. So it's like, you have your connections depend on like the structure of the tensegrity. So like if you have things that are like if you have maybe things that are holding in tension, you might have like connections that are sort of symmetrical in nature. So you wouldn't have the conventional random scale-free structure. You'd have something else. And that would actually be interesting because it's a very different type of network, very different type of structure, and things propagate through those networks differently, actually. And I don't know
if they're have let's see, high degree of elasticity, like, or if they're stiffer, you get more propagation through the network than if you've got something that's soft elastic.
Right.
Then it tends to stay in the general area, but if it's tension element, which is elastic, and it's say like a string that is not elastic, you get the forces move throughout the whole network.
Yeah. Yes.
Anyway, I can demonstrate that one. A model I have here.
Oh, wow. So the edges would be, in that case, the edges would have different properties as well, where you would have some that are like, have a certain impedance, and you'd have to define that. So it's interesting that you can, a lot of the complex networks research that's been done, they kind of assume the edges are uniform. Maybe they have a weight, but that's usually the connection weight. They don't think about the physics of the connection usually. And so if you have a soft material, that edge actually maybe is weighted by physics in that maybe like a correlation or something like that, which is usually the case. I guess you could use a correlation, but it wouldn't mean the same thing, quite mean the same thing.
Well, anyway, the physics of tensegrities gets complicated.
Yeah.
In a hurry, especially if you're perturbing them with a frequency, you can get them to collapse.
Right. Yeah.
Yeah. Anyway.
Yeah.
Sometimes you want that, and sometimes you don't.
Yeah.
And if you're using COMSOL, it thinks you're building a bridge. So if it collapses, this is a bad thing, and it won't produce an output. It just says error. And really, no, that's not an error. It's fine if it's kind of collapsed. That was fine. That was what I wanted.
Yeah.
Anyway.
Well, if a cell collapses, that's a bad thing too, right?
It's held up by its neighbors.
Oh, okay.
So
that's true. Yeah.
It's networked in that way.
Yeah, it is.
And when it collapses, they turn into a mesenchyme cell that goes and crawls off somewhere. And they can move as a cell sheet as well. There's the 3 different modes.
Yeah.
But they're literally held up by their neighbors. They're a network that way.
Yeah.
So
what happens?
Just trying to get your head around it. Just
So one aside on that, what happens if you have like apoptosis or something like that, where the cell dies in a sheet, and do they just kind of fill in? I mean, from a structural perspective, I guess.
They squeeze it out. And there's images of it being squeezed, and it goes popping out the top, and then there's sort of a hole, and then it fills that in.
So,
and it produces a different patterning in the cell sheet, and then that resolves itself as well. Like I said, things like T1, T2, T3 transitions gets into that area of things. But yeah, literally it squeezes the apoptotic cell out the top, and it goes springing out. Apparently, it really does act like a spring.
yeah.
It expels the dead cells.
Wow.
The network does.
And so the network is able to maintain its stability throughout all this, right? There's no.
Yeah, it does. There's a rosette configuration that's formed when that happens, which is interesting. It tends to cause a rotation in the cell sheet. And
so if you stuck like a little probe, like a pipette in there, you couldn't like disrupt that process. It would just kind of be stable to that perturbation.
I don't know. People have ablated holes in cell sheets and looked at the forces, and the cell sheets just tend to fill in the hole. Wound healing,
Yeah.
Yeah. I don't know. I'm trying to understand it more.
Yeah.
Sort of, and I just discovered that there's a paper that's by Fredberg and starts with an A. Do I have it here? I'll send it to you.
Okay, yeah, that'd be good.
Because it's a history of their lab and how they've been working with epithelial cells and still haven't figured it all out. But it's a very nice summary of the study of epithelial cells. So anyway, yeah, it's Fredberg.
Okay, yeah.
I met him one time. Came to the university.
Yeah, that's great. Okay.
And, need to get you that. Huh?
Yeah. So, did anyone else want to say anything before we go for today?
I also have some idea for some work if you actually if anyone would be interested. Following my conversation at the beginning of the session, I've been trying to actually have some comparison between artificial neural networks and intelligent networks. And so I've decided to write the first chapter of the book I was talking about. This is an introduction that is going to discuss how historically actually development of the brain of the C. elegans is actually what has been studied so far from a developmental point of view to the nervous system of C. elegans from the stage, maybe the first stage of its development to actually maybe 21st or 20th, for example, second day of its life. So I would be happy if anyone is interested in collaboration with this review paper. Maybe I'm going to more focus on how actually different works have been done so far on the different from the simplest model that has been so far discovered from how the C. elegans is working. I mean, the mathematical model mainly, to maybe the most complex one so far. So if you know of some maybe good references that we could start with, I would be happy to start with some references and then add them to the main review paper or the main chapter, and I'm going to discuss it every week of my progress, and so would be happy if anyone is interested in collaboration on this chapter or maybe review paper as well.
That sounds great, Vahid. Yeah, I'm sure that we have people interested in that. So, yeah, and I'll send you the things that we were doing on that. I mean, C. elegans is an interesting case because we know so much about the nervous system. So that sounds like a good place to start.
Okay, the reference is in the chat.
Okay, yeah, I saw that. That's Biophysics Reviews, yeah. Okay. Yeah, thank you for that. A life off the beaten track in biomechanics. Yeah, great stuff. Okay, well, thank you everyone for attending. And see you next week.
Okay.
Thank you.
Thanks, everyone.
Thanks.
Bye.